there, so this is topic 20, biotechnology and genetic engineering. These are the objectives we have to look at. And this is the first objective we're going to look at today. And we have to state how bacteria, the biological organism, are useful in genetic engineering and biotechnology. First of all, what are these things? Biotechnology is basically using living organisms, biological organisms, biotechnology, in our technology, in industry, to create different things. Genetic engineering is using those organisms but manipulating their DNA, their genes, genetic engineering, to create products that we might need. Examples are antibiotics and insulin. Insulin we can use bacteria for, and antibiotics we can use fungi to create. So bacteria are used in both processes, and the reason for that is because they reproduce really quickly, so they can make a lot of the product really quickly for us and save us a lot of money. Other reasons, so objective B is why they're useful, the bacteria, and other reasons are basically there's no ethical concerns about using bacteria. Sometimes cows are used to make milk with antibiotics in, and this can sometimes harm the cows because they're given hormones to grow quickly and produce a lot of milk with antibiotics in. So there's ethical concerns there, but with bacteria, not so many. So yeah, it's quite easy to do. Their genetic code is very similar or the same as all other organisms. So if you wanted to create, let's say, some insulin for a human or any other animal, you can quite easily take a bit of the DNA from the human and or other organism and place it into the bacteria. Their genetic code is very similar, if not the same, to other organisms, so it doesn't normally reject the code and incorporates it into its own. And if you don't know already, the DNA itself is coding to make different things. So if you wanted to make some insulin, you could pop the gene from the human or whoever that makes insulin into the DNA of the bacteria, and it would make the insulin for you. So bacterial DNA, it's two types. One type is just the circular strand of DNA, no nucleus, just floats in the cytoplasm, and then you've got these circular things called plasmids. Now, plasmids are the important thing for genetic engineering, because what happens is the scientists would cut open the plasmid and then take the gene that would create insulin, pop it into the plasmid, and then as the bacteria itself reproduces and makes more bacteria, the plasmid would reproduce as well, the new modified plasmids. So as the bacteria reproduces, it will make more bacteria with this new modified plasmid in, and that new plasmid would code for making insulin. So you'd have all these different bacteria making insulin, and a lot of it. All you have to do then is extract the insulin. Extraction usually ends with the destroying of the bacteria, but again, there's not a lot of ethical concerns there. So that's the end of 20.1, biotechnology and genetic engineering. Okay, the second part of this topic is looking at more detail into biotechnology. It's 20.2, those are the objectives. And first thing here is to look at anaerobic respiration, which is without oxygen, and how that's used by yeast to make things like ethanol and biofuels. Now, the process that it's called is fermentation. That's when you use respiration of things like yeast or bacteria to make different products. It's called fermentation, and it's a type of biotechnology. So brewing beer or wine using yeast, or baking of bread using yeast, or making cheese using bacteria. Basically what you do is you get the organism, use a food source for them, they use that food source for respiration, create some byproducts, and those byproducts that they make are the things that you're collecting, like ethanol or biofuels. Another type of biotechnology is antibiotics, penicillin. Now that's made using a mold, which is fungi, and it can be made using bacteria as well. Now, citric acid and lactic acid, it's that chemical that you make in your bodies all the time, lactic acid, when you anaerobically respire, i.e. you don't have enough oxygen when your muscles work, so that it creates lactic acid as a byproduct of not having oxygen available for respiration. Bacteria and fungi can do the same thing, and they create lactic acid or citric acid off the back of that. Sewage disposal, we will have talked about that in topic 19, ecology. It requires bacteria on filter beds to purify the waste. They use that the sewage waste to respire. Okay, so fermentation in a bit more detail. Microorganisms like fungi or bacteria are going to use this process for respiration to produce energy for their living things. Okay, now they can do that with or without oxygen. Aerobically is with oxygen, and anaerobically is without oxygen. Microorganisms will be given glucose, salts and vitamins to encourage their growth. You'll also need to control the temperature and pH as well. Okay, so one thing that can be made by fermentation is biofuels. Now, biofuels can be either ethanol or oil, and these can be used in cars. Ethanol can be used with or to replace petrol, and then your oil can be used to replace diesel fuel. Now you'll probably want to relate this part to the renewable and non-renewable section in topic 19. So sugarcane you would give to the microorganism, that would be the food source, and they would use that food source to create ethanol by fermentation. And then the ethanol can be used to replace petrol. Rapeseed oil or rapeseed oil, I don't know, or sunflower seeds, they are given again to the microorganism as a food source, and they use fermentation, respiration, to create oil. The oil that can then be used to replace diesel fuel. The reason we do things like this is A, petrol and diesel are quickly running out, so we need to find alternative fuels, and they're also less polluting. The issue with it is that they're quite expensive to produce. You think about all the technology and procedures and manpower that's got to be put in place to get a whole bunch of yeast or whatever to start a fermentation process. It's not yet economical to use, and that means basically it's not going to make us a lot of money. It's going to cost us money to make, and these are some of the issues that are outlined with that. It costs a lot of money to grow the sugar cane or the sunflower for the seeds. Fertilizing them, harvesting sugar cane is expensive. Also, when you extract and convert the sugar into ethanol, that uses quite a lot of energy. It uses more energy than is actually released when you burn the ethanol later on to use in the cars. So you're actually expending more energy than the ethanol that you're creating is going to make for you. So it's a bit counterproductive almost. Deforestation's got to happen to make the land for the agriculture, so you've got all the issues there with loss of habitats, global warming, etc. However, you know, it still stands that it is renewable and it's less polluting. The next thing to talk about, the next objective is how anaerobic respiration is used in yeast for bread making. So the first one was about how we use anaerobic respiration for making like ethanol, for biofuels. This is about bread. So it's made using yeast, which is a fungi. Now, what happens is, off the back of the anaerobic respiration, CO2 is made. Now, CO2 in bread would bubble up and create a light and fluffy texture within the bread, which is why they use yeast to do it. They give certain ingredients like salt, oil, flour, water, and then obviously the yeast. Now, yeast doesn't have enzymes to digest the starch in the flour, because the yeast is going to need glucose to carry out the fermentation process, because it needs that food source. But there is no food source here because it can't break down the flour because it's made of starch. Luckily, flour actually has some amylases inside the flour that will break down the starch in the flour to glucose, so then the yeast can actually use it. So yeah, quite a useful process. You can make bread off the back of it, and it's using anaerobic respiration, which means without oxygen. The CO2 is useful because it creates bubbles. Okay, so this is the process in a bit more detail. The yeast, like we said before, will use or ferment the sugar during respiration, and it will create alcohol, and CO2. Now you might be thinking at the minute, hang on, bread's got alcohol in it. It obviously doesn't, and we'll explain why later. The CO2 causes the dough to rise and create that light and fluffy texture. Now, you might think, why don't the bubbles just disappear? Like bubbles would disappear from a milkshake. The bubbles travel to the top and then they pop. Why does that not happen? Well, basically there's this protein in bread called gluten. It gives the bread a sticky texture when it's baking. I don't know if you've ever made bread, but when you do, the flour it becomes very sticky when you start to make it. That's the gluten is in the flour. And what happens is when the CO2 is made, they start to bubble up and then they're caught and held in place by the gluten. Kneading is the process where you start rolling and folding the bread, and it adds more oxygen into the bread, and this can be used for respiration by the yeast. It's heated at 27 degrees initially because you want yeast to respire, and if it gets too hot, the yeast will denature. So you want the enzymes to be able to work as well. So keeping it at that 27 degrees Celsius temperature is good because it allows the yeast to work to make the bread rise, and it allows the enzymes to break that starch down into glucose for the yeast. However, after that, you then heat the bread at 200 degrees, and this is why. You want the bubbles to expand, so it makes the bread even bigger. It kills the yeast and causes that alcohol that was originally produced to disappear. It evaporates. At that point, the dough that's got a few bubbles in it will turn to bread. Okay, objective C is to describe and use an investigation about pectinase, which is an enzyme in fruit juice production. We'll come to that later. First of all, there's a couple of things to talk about. First thing is how we can make enzymes like pectinase for use in industry. So again, we're going back to our little friends called bacteria that we can use to make said enzymes. So what happens is, it's all written here, but I'll go through the picture. You put in here a feedstock with some bacteria. So the feedstock goes in here, fills up this tank, and the bacteria are going to be in here as well with the feedstock. In there you've got the nutrients available for bacteria to carry out respiration. You're going to put food in here for the bacteria, and what they're going to do is digest that food. And what do you think they make to digest that food? Well, it's enzymes. Now they make two different types. They make an intracellular enzyme and an extracellular enzyme. The intracellular enzymes, let's start with the extracellular enzymes. The extracellular enzymes are those enzymes that are made by the bacteria and then secreted out of the bacteria into this fluid to break down certain chemicals like nutrients into things like glucose or whatever, so they can be absorbed. Some things will be absorbed by the bacteria, and the bacteria has its intracellular enzymes to then break them down further. What happens then is you've got loads of bacteria with the food that's diminishing, and you're going to have a lot of enzymes made. This then, this whole feedstock material with the bacteria and the enzymes travels through this tube. Before we talk about that, I want to talk about the paddles and the air here. The air, which has oxygen in it, is pumped into this feedstock to give the oxygen to the bacteria for respiration, which is important, and the paddles there to stir it to mix in the oxygen with the bacteria so that the process happens quicker. Bacteria respiring, they're going to make carbon dioxide, so there's a little vent here for the waste gases. Now, as we said before, bacteria make enzymes, two different types. This whole sludge is going to pour through here, and it's going to lay it onto this filtered bed here. Now, if you're an extracellular enzyme, you're in the fluid, you're floating, and you're small enough to fall through this little filter tray into the bottom here. So the extracellular enzymes are extracted as they fall through the bed. They're small enough. However, the intracellular enzymes in the bacteria are not small enough to fit through. So they're carried off this way, and then you have to then basically destroy the bacteria's cell walls to get into the enzymes to extract the bacteria, so there's an extra process there. Now, genetic engineering. You can do a little process where you add some genes to the bacteria so that they create certain enzymes that you want, like pectinase or whatever, And you can make them more heat resistant, because sometimes enzymes are used in washing powders, and washing powders need to work at high temperatures sometimes. So if you can make the bacteria more, the enzyme more heat resistant, then that's really useful. Immobilized enzymes, we've got to say what they are and why they're useful. What they are is enzymes that are immobilized. They're not allowed to move, and so basically they're held on beads of membranes or whatever in a tube, and then the stuff that they're going to react on or digest or whatever is poured through it. Now they're kept on these membranes that are insoluble. The enzyme beads are kept on these membranes, and the reason they're insoluble and unreactive is so that the enzyme doesn't react with it, and they just react with the substrate that's going to be poured through them. So these enzymes, which are on the membranes, are packed into columns, and the substrate's poured over them, and then they react and break down the substrate and create the products, and the products fall through the end of the column. The reason this is good is that the enzymes aren't in the mixture, so it's not lost every time the product is extracted. So what you would do is, here you could create your enzymes using the bacteria, and then you could take these enzymes and fix them onto beads, so that when you use them in the reaction that you're going to use them for, you don't lose them. They're not going to fall into the product and then have to be extracted. So advantages are basically that the enzymes are not lost every time the product is extracted, and it allows you to make that product continuously rather than by batch. By batch, it means you'd have to reset it all up, whatever process you're doing, and you'd have to replace the enzymes. So immobilized enzymes are really good. So this is what enzymes are normally used for. Washing powders. Remember that enzymes, you will have done this topic before, they basically either build things up, which is anabolic enzymes, or break things down, which are catabolic enzymes. They're used in washing powders, obviously, that's a catabolic one. They're going to break stuff down like stains. Hair removal, same thing. Cheese manufacture and tenderising meat. So proteases, all of these proteases. Lipases are used in flavour enhancers, in cheese, or washing powders. Pectinases is the thing we're going to talk about soon, is used for juice clarification and juice extraction. Basically making pure juice from a sludgy fruit mixture. Amylase, they can be used to produce glucose from starch in things like flour. Pectinase in more detail then. Separates the juice from a fruit. Now pectinase can be extracted from fungi. So you get a bunch of fungi, the microorganism, you can take the pectinase from that. Now they break down a thing called pectin in fruits. Now pectin is in a fruit, and it's a jelly-like substance that sticks the cell walls of the fruit together. What happens is you would take the fruit, create a sludge with it, so you'd blend it up, and then you would add some pectinase, and it would break down the pectin in between the cell walls. And during this process, you would release a lot of juice from the liquid of the cells. However, many polysaccharides, long-chained carbohydrates, are released during this process, which makes the juice very cloudy. Pectinase can then be used again. It can make the cloudy juice less cloudy. It can clarify it, make it more pure. And we use that for things like, if you looked at apple juice that you sometimes get from restaurants made pure, it's very cloudy and thick and fills you up. Whereas your juice in your supermarkets that's made from a concentrate has had pectinase to clarify it, to make it clear, and it's not as filling. Wine, again, you purify it using pectinase, you clarify it. Pectinase also breaks down the large polysaccharides into smaller sugars, and smaller sugars obviously make the juice sweeter. So pectinases are really useful, as they can extract juice, they can clarify the juice, and they can make the juice taste sweeter. So there is a practical to look at regarding the use of pectinase in making fruit juice. There's a lot of detail on the PowerPoints, but basically you set up equipment where you've got a funnel full of apple puree, thick apple puree, and you add some pectinase to it. You have that funnel sat in a measuring cylinder, and what's going to happen is the pectinase is going to work, it's going to break down, it's going to extract the juice from the puree, and that juice is going to drip down into the measuring cylinder. All you do then is you set up that same experiment a few times at different temperatures, because enzymes work better at different temperatures. The enzymes will work really slowly and not a lot of juice will be made at low temperatures, but at high temperatures, like maybe 30 to 40, the enzymes will work a lot quicker and you'll make a lot more juice. After 50 degrees Celsius, the enzymes will be denatured and won't make any juice. So that's basically it. There's a couple of things to add. First of all, the juice has been extracted and clarified so that the juice in the bottom of the measuring cylinder will be clear. You could go a bit further and say, hey, why not, instead of using pectinase, why don't I use cellulase? Cellulase breaks down cell walls. Will that work? What about amylase? Amylase breaks down starch. Will that work to extract juice? You could use those and see which is the best. Maybe a combination of both. We're not going to, but it's just further thinking because they might bring it up in the exam. So the last thing we're going to talk about on this video is the use of enzymes in biological washing powders. We'll do a little investigation as well. Now, back in the day, people used to wash their clothes in warm water, and you had to get the temperature pretty high because you can remove some, you can break down some chemicals using high heat energy. The particles vibrate and become loose, and then that's how people wash their clothes. Obviously costs a lot of energy because you've got to heat the clothes up really high, and damages the clothes as well because the clothes are made of chemicals, right? So when they made, because those were washing powders, and when they made the biological washing powders, which had the enzymes in, that changed everything because now you can wash the clothes at a lower temperature because the enzymes do all the work rather than the heat energy breaking down the chemicals. So the enzymes, the main ones that are used are proteases and lipases, so they're breaking down things like proteins and fats like blood, eggs, gravy, or fat like grease, or, you know, the yolk of an egg is also fat. So these are stains on the clothes, and what happens is when you put them in the warm water, only warm, needs to be warm, for the enzymes to work at an optimal temp, the stains are digested into small soluble molecules which can pass out of the cloth, and then you wash your clothes effectively. They save energy, save money, because you can wash at low temperatures, and you only need to put your washing machine at 30 or 37 rather than 80 degrees. Problems: if temperatures in the washing powder, in the washing machine, are too high, the enzymes will denature. If you think back to where we talked about how we make enzymes, i.e. using bacteria, we can use genetic engineering to make the bacteria create enzymes that are more resistant to temperature. So the investigation involves eggs. You take some egg, you've got the yolk and the white, which are the white bit's protein and the yolk bit's fat and protein. So you stir it all up, and then you add it to 4 pieces of cloth so that they're now stained, and you leave them to dry, and you're going to put them in 4 different beakers, beakers A, B, C and D. Beaker A is going to have warm water in it, which is good, we like warm water, but it's going to have no washing powder. The next beaker is going to have the same setup, but instead of no washing powder, it's got washing powder. This washing powder is non-biological, which means no enzymes, so it's just the washing powder, and again it's in warm water. C has got the biological washing powder, this is the good one, and it's got the warm water, and then D has got the biological washing powder again, exactly the same, but it's been boiled for 5 minutes. So what happens in the end is this one, A, doesn't work, it doesn't remove the stains. The stains are very strong fat and protein stains. Without washing powder, you're not going to remove any. B works a little bit. The washing powder itself has got emulsifiers in there and chemicals that can break down some of the stains. C works the best. It's got enzymes in that break down the protein and fat stains, and they become small soluble molecules like amino acids and fatty acids and glycerol, and they just dissolve into the water away from the cloth, and it becomes stain free. D has got the enzymes, but they've been denatured by the boiling water. So again, it doesn't work. So what we take from this is obviously the results as standard, but the control. Which one was the control? The control was the one with no washing powder, because you're trying to test whether enzymes work better, so having the non-biological and biological is your experiment. You then throw an extra one in and say, well, what if we boil it to see how a temperature affects it? But the control is the one with no washing powder. Okay, so that's the end of that video. Thanks for listening.